All right. Thanks everybody for coming to what I think is the first Masonic Cancer Center seminar of the semester or the school year. And I'm very pleased to be introducing my friend, colleague, and mentee, Dr. Adam DeSmith from the University of Southern California. For those of you who haven't heard him speak before, you'll soon learn that he was raised in the UK, where he was trained at uni at the University of Bristol, and then got his PhD in genetics from Imperial College London, and then he hopped over the pond to the Bay Area and did a postdoc with Dr. Joe Wiemels at University of California, San Francisco, where he quickly established himself as a pediatric leukemia researcher, especially taking great advantage of California's resources for conducting these studies in population-based way. And then several years ago followed Joe, but standing very much on his own, to the University of Southern California, where he is faculty in the Center for Genetic Epidemiology. And really, I have to say that Adam is the single best molecular and genetic epidemiologist of ALL of his generation, working in the world today. So I'm very pleased to call him a collaborator and excited about his talk today. So Adam, take it away.
Thanks, Logan. That's far too generous. And I wasn't nervous before, but now I am. So yeah, very grateful for the invitation to speak to you all today. It's nice to come back to Minneapolis and St. Paul, especially in this time of year. So today I'll be talking about some of my research into trying to understand what causes childhood leukemia, as well as some of the disparities. So I'll briefly talk first about what we know about the epidemiology of childhood leukemia, and then what I think is a neat story about how a novel variant in the IKZF1 gene contributes to the increased risk of ALL in Hispanic Latino children. Then some ongoing research as part of the RECORD study in collaboration with Logan, trying to understand the early life origins of both childhood ALL and AML. And then finally, an example of how we can use tumor genetics to inform etiology at the end here. So I probably don't need to introduce childhood leukemia to most of this audience, but first of all, just to highlight some of the differences in the age at diagnosis. So this is childhood ALL, where we see a peak age of incidence between the ages of 1 to 6. Just to note, we also see a male sex bias for ALL. In AML, we see actually this increases as we get older. But in children, we do see this slight peak in very early life. And so these early life peaks of incidence suggest in utero origins of childhood leukemia. And this peak for ALL is also thought to be related to the delayed infection hypothesis. So this was proposed by Sir Mel Greaves back in the late 1980s, that children who are not exposed to enough infections in early life have dysregulated immune development and have a higher risk of developing leukemia in the face of infections later in childhood. So in terms of the natural history of childhood ALL, we know that often there's an initiating lesion that occurs prenatally. So this has been shown from studies of twins, where both twins have been found to have, identical twins have the same exact fusion breakpoints, and also through backtracking studies where people have used neonatal samples, mainly blood spots, to look for mutations that they found in the leukemia samples and found that they are indeed present and detectable at birth. But we also know that not all pre-leukemic clones will become overt leukemia. They require these second hit mutations, including point mutations and deletions of genes involved in cell cycle control, B-cell differentiation. So we think there may be genetic and environmental factors that influence both the development of these initiating lesions and of these second hit alterations. And for those of us who are interested in what causes ALL, we're starting to think about the potential for newborn screening of pre-leukemia for earlier detection and perhaps even prevention of childhood ALL. And I'll come back to these points later on. One of the most striking trends in the epidemiology of ALL is this increased risk in self-reported Hispanic Latino individuals. So this was an analysis of SEER data from a few years back. We see actually the increased risk of ALL in Hispanic Latinos is consistent across all age groups. And you can see here actually non-Latino Black individuals, particularly children, have the lowest risk of ALL. And Logan is leading a study trying to understand that difference. Interestingly, this increased risk is driven by the B-cell immunophenotype. And this suggests that there may be some biology that's underlying this health disparity. And the germline genetic contribution to ALL risk is well established. So up until now, genome-wide association studies, or GWAS, have identified at least a dozen well-replicated loci. Many of these genes are involved in hematopoiesis and B-cell development. And noticeably, several variants have an increased risk allele frequency in Hispanic Latino populations compared to European populations. So these numbers are from the Genome Aggregation Database, or GNOMAD. And just to highlight a couple of these, so ARID5B and GATA3, you can see quite considerably larger risk allele frequencies in Hispanic Latino populations than Europeans. Same for GATA3. For ERG, we actually don't see really a difference in the risk allele frequency, but we did see a difference in the effect size, suggesting that there may be gene-gene or gene-environment interactions driving this. And I just wanted to also highlight IKZF1. This was the first SNP discovered in the IKZF1 gene. You can see the risk allele frequency is maybe slightly higher in Europeans. And I'll come back to this gene in a couple of slides. So also in our relatively recent multi-ancestry GWAS using California-based ALL cases and controls, we constructed a polygenic risk score. And we could see that the PRS, and this was just using 18 of the known SNPs, this was shifted to the right in Hispanic Latinos compared to non-Hispanic whites, and highly significantly different. And we even see this when we stratified by case control status, that Hispanic Latino controls have significantly higher PRS than non-Hispanic white controls. So about a 10% increase in the mean polygenic risk score, which may partly explain the increased risk of ALL, although this is simplifying things a little because there's questions over how appropriate it is to compare polygenic risk scores between populations. But beyond the genetic loci that I've mentioned, are there additional factors that may contribute to the increased risk of ALL in Hispanic Latinos? So that brings me on to this project, and over the next few slides, I'll present what I think are some exciting recently published work on the role of IKZF1 or Icaros genetic variation on ALL risk in Hispanic Latinos, and I'll discuss some of our ongoing research. So IKZF1 or Icaros is a transcription factor and a critical regulator of early lymphoid development. The loss of IKZF1 and other key B lineage factors blocks development of B cells at a stage of differentiation when they have a high proliferative potential and may be more susceptible to malignant transformation. We know that somatic loss of IKZF1 is common in B-cell ALL. So about 20% of the tumors have loss of this gene, or at least one copy of this gene is lost. And it's enriched in certain subtypes like Philadelphia chromosome and PH-like ALL that are associated with poor prognosis. We also know that rare germline variants in IKZF1 predispose to both immunodeficiencies and B-cell ALL. And as I've mentioned, GWAS have found associations of IKZF1 with childhood ALL, but also other phenotypes, including blood cell traits, allergies, and autoimmune conditions. So it's clearly a gene of interest, but the number of the common IKZF1 risk loci for ALL, the causal variants, and their underlying mechanisms had not been comprehensively studied. Previous studies, including our own, some of our own and from other groups, had identified at least two independent risk variants or risk loci at IKZF1. but there had been, as I've said, no comprehensive studies. So we had conducted this multi-ancestry GUS of ALL in the CCRLP, the California Cancer Records Linkage Project. And this was, the cases were identified through linkage between the California Biobank Program and the California Cancer Registry. So we had about 1,900 Hispanic Latino cases, over 8,000 controls, about 1,200 cases in non-Hispanic whites, and a very small study in East Asians. What we did was we performed what's known as conditional association testing across the IKZF1 region, and we wanted to identify the total number of independent risk variants, try to identify the causal variants through fine mapping, and possibly the cell types in which they operate and underlying mechanisms. First, we know that Hispanic Latinos are a highly heterogeneous group, so culturally, phenotypically, as well as being genetically diverse, and individuals exist across the spectrum of each of these. And Hispanic Latinos are genetically admixed, comprising European, African, and Indigenous American ancestry. So this is showing data from the 1,000 Genomes Project, where these 4 populations are all described as being Hispanic Latino. You can see clear differences. For example, the Peruvians have much higher contribution of Indigenous American ancestry, whereas Puerto Ricans and Colombians have more of a contribution from African and European ancestry. And by studying genetic ancestry and the variants associated with ancestry, this may provide clues to the increased risk of ALL in Hispanic Latino individuals. This is showing a similar plot for our CCRLP Hispanic Latino cases and controls. So you see a similar spectrum of ancestry, European, Indigenous American, and African. And just to point out that the race ethnicity groupings in our study were based on self-reported data. So we did not reassign anyone based on their genetic ancestry. So using our CCRLP GUS data, as I said, we performed this conditional association testing. So first, just to show our initial GUS Manhattan plot, we could see what looked like 2 peaks at each end of the IKZF1 gene, one in the promoter region here and one just in a downstream region. When we adjust for this top SNP, and that's what this conditional association testing does, when we throw that SNP into the model, we see that first peak disappears, and we see that this downstream peak remains genome-wide significant. Now, if you look closely, you might see maybe there's some other SNPs of interest down here. So when we adjust for both of these top SNPs, we then see this third independent GUS peak remains, or actually jumps up to genome-wide significance. So this is in Hispanic Latinos. And when we looked in non-Hispanic whites, we actually only see 2 peaks. So we see a peak at the promoter region and the downstream region. And when we adjust for that top SNP, that one goes away and we see this promoter region peak. And then when we adjust for both of those, we don't see any more peaks. So we think that Hispanic Latinos are unique in having 3 independent risk loci at IKZF1. I'm not showing the East Asian data, but it shows a similar story. So this is the novel risk allele. Let's call it SNP3. You can see it has about a 20% risk allele frequency in Hispanic Latinos in our data, and about 1% in the non-Latino whites. If you look in NOMAD, it's actually lower than that in European populations. So why does it have such a high risk allele frequency in Hispanic Latinos? Well, we show that the risk allele is both associated with global Indigenous American ancestry, and that frequency of the Indigenous American haplotype at this SNP was higher in cases than in controls. So we would have actually discovered this locus through admixture mapping. So we think that the high risk allele frequency at this SNP is attributed to their Indigenous American ancestry. So we next looked at ancient genomes to see when the allele may have arisen in history. So we analyzed shotgun sequencing data from individuals across the Americas. And what we found that the oldest sequenced Indigenous American individual actually carries this risk allele in IKZF1. And it's present at a relatively high frequency among both ancient and modern individuals through the Americas. So the green color here means the allele is present, and the darker the color is the older the individual that was sequenced. So it seems that the risk allele is at least around 13,000 years old and seems to have spread quite quickly through the Americas. So the next obvious question is, why might that have happened? So we explored signatures of positive selection at the IKZF1 locus across populations and found significant selection in Hispanic Latino populations, but not in European or an East Asian population. And East Asians actually have this risk allele at about 20% risk allele frequency. So altogether, what we think happened is that the allele arose sometime after the split of Europeans and Asians. And then entering the Americas, there was likely a reduction in the frequency of this risk allele due to a population bottleneck. And that since this has increased, and in modern day indigenous Americans, this allele is actually found at about a 50% frequency. So then through admixture between indigenous Americans, Europeans, and Africans, we see about an 18% risk allele frequency in modern day Hispanic Latinos. So maybe if I'll come back to this at the end, some potential reasons as to why we think this allele may have been selected. So to recap, there are 3 independent risk alleles at IKZF1 in Hispanic Latinos, one of which is at the promoter region, and the other 2, including the novel one, SNP3, have putative causal variants located within a regulatory region. And I'm not going to go into the details, but through statistical fine mapping, we actually pinpointed down to 2 causal variants that are 26 base pairs apart for these 2 independent loci. And they're actually on opposing haplotypes. And we looked at promoter capture high C data from human lymphoblastic cell lines. And this shows that this regulatory region shows a strong interaction with the IKZF1 promoter. Oops. So we also looked at expression quantitative trait locus data. This was in collaboration with Dr. Linda Kachuri at Stanford, who had analyzed whole genome sequencing and RNA sequencing data from about 2,000 Hispanic Latino children. And this did show that both the promoter region SNP and the SNP3 were associated with reduced IKZF1 expression in these Hispanic Latino children. We then looked at chromatin accessibility at these regulatory regions using single-cell ATAC-seq data, covering human hematopoiesis, covering cells across the entirety of human hematopoiesis. And it actually showed that there appears to be an enhancer that's enriched in the pro-B cell type. And this finding is consistent with the fact that IKZF1 SNPs appear to confer risk for B cell ALL and not for T cell ALL. So as I've mentioned, these putative causal variants are 26 base pairs apart. They reside in this enhancer region that is most active in pro-B cells and regulates IKZF1 expression. And just to show some of the single-cell ATAC-seq data across more cell types, it really shows how specific this enhancer region is to early B cells, pro-B cells, and common lymphoid progenitors specifically. So we did do some more kind of cool functional work, mainly in Dr. Vijay Sankaran's lab, who was a collaborator on this work. So if you're interested in that, you can look up our paper. But just to summarize, we've shown that Hispanic Latinos are unique in having 3 independent risk loci at IKZF1. The novel risk allele has about a 20% frequency in Hispanic Latinos. It's basically absent in Europeans, shows evidence of positive selection, and we think contributes to the increased risk of ALL in Hispanic Latino populations. The Hispanic Latino enriched risk allele appears to reduce enhancer activity, chromatin accessibility, and IKZF1 expression, we think likely by disrupting a positive autoregulatory mechanism. So I didn't show this, but it does disrupt an IKZF1 binding motif, and we showed that IKZF1 does bind to this enhancer region. But the precise impact of this variant and other IKZF1 variants on ALL risk is to be determined, but we think is likely through stalled B cell development that then leads to increased chances of additional mutations occurring in pre-leukemic clone. So another outstanding question is, how might this SNP affect patient outcomes? So hopefully we'll be able to get some data on that one day. Some more outstanding questions in general in terms of the disparity in ALL risk. How much of this disparity is due to genetics versus the environment, or a combination of the two? What's the contribution of rare germline variants? What factors may underlie the rising incidence of ALL in Hispanic Latino populations? So I didn't show this, but ALL incidence is increasing only in Hispanic Latino populations, at least in the US, so, which is unlikely due to our genetics. And ultimately, how might we be able to reduce ALL risk in predisposed individuals? So part of my research program is focused on answering this first question. I think it's much easier than trying to answer this last question. So I was fortunate a couple of years ago to receive an R01 grant to study, do a kind of comprehensive genetic study of ALL risk loci to try and understand the genetic component of increased risk in Hispanic Latinos. So we're putting together all of the available GUS data sets, so in collaboration with Doctors Philip Lupo and Michael Scher at Texas Children's Hospital, June Yang at St. Jude's, and Catherine Mataya at UC Berkeley. So we have upwards of 5,000 cases, about 30,000 controls. We're also going kind of beyond GUS, doing admixture mapping, transcriptome-wide association study, or a TWAS. And then we want to develop polygenic risk scores that might best predict ALL risk in Hispanic Latino children. And then also incorporate both genetic and non-genetic risk factors into G/E studies, including a polygenic risk score as the G. And then we also received this supplement to genotype an additional 650 cases and 200 controls from a Mexican ALL study. So this is in collaboration with Dr. Juan Manuel Mejia Aranguren. And this, as well as just increasing our sample size, I think will be really interesting to look at the effects of ALL risk alleles and performance of a polygenic risk score in a Hispanic Latino population outside of the US. And I should say that these data will also contribute to a global effort led by Logan and others for the childhood leukemia, what is it? Childhood Cancer and Leukemia International Consortium, or CLIC, which is putting together all of the existing data on ALL from around the world. Just to show some preliminary data from a meta-analysis of existing ALL GWAS data in Hispanic Latinos. So we have almost 4,000 controls. So we found, some of our well-known genes. And if you look closely, there's something on chromosome 5 that almost but didn't quite reach genome-wide significance. But hopefully with additional data it will. But if we zoom in on the region, it's really interesting in terms of the genes that are in this region. So there's a DNA repair gene, RAD50, which is part of the NBN complex. There's also interleukin genes, including IL-13, which is a cytokine involved in allergies, infection, and B-cell proliferation. Also, I think interesting that one of the top SNPs, actually all of the SNPs have a much higher frequency in Hispanic Latinos than Europeans. One of the top SNPs has about a 5% frequency in Hispanic Latinos, but is basically absent in Europeans. And we've looked, and it is associated with local ancestry in our data. So part of the next steps are to replicate, or at least add more data to our GWAS to push it over the line, and then do some functional analysis. So I've been fortunate to collaborate with Dr. Vijay Sankran's lab, and we recently received an R01 as MPI. Well, on the next slide, or in a couple of slides, I'll talk about what we're doing. But this was a paper we published a couple of years ago where we integrated GWAS data with some single cell, this was ATAC-seq data from kind of the trajectory of B cells from hematopoietic stem cells to mature B cells. And then we looked at the enrichment of ALL risk alleles at these different stages, and we could show that childhood ALL GWAS SNPs are enriched in the kind of pre-B, pro-B cell types. So as well as kind of identifying the B-cell stages that genetic risk alleles appear to play a stronger role, This is perhaps pointing to biology of the disease. We also can use these data to try and identify causal variants. So by identifying enhancer regions that are most enriched in these relevant cell types, for example, at CEBPE, we confirmed what had been proposed to be a causal variant in the promoter region or upstream of this gene in a region with chromatin accessibility that was kind of most active in pro and pre B cells. So next, what we want to do with this R1 is to follow up on these results to understand the function of B cell ALL risk loci. So again, we're integrating our multi-ancestry GWAS through fine mapping, and then integrating that with single-cell RNA and ATAC-seq data, spanning human hematopoiesis and B cell development differentiation, to try and identify these B cell developmental stages and cell states that underlie the B cell ALL predisposition, and to try and identify the causal variants and target genes. And then what I think is most kind of cool about this is, and this is all Vijay's lab, doing genome editing to actually introduce what we think are the causal variants into hematopoietic stem cells, and then looking at their effects on B cell differentiation and development, and also looking at the effects on illegitimate VDJ recombination or RAG recombination. And we're going to be prioritizing 6 causal variants, including ones that we think are involved in the increased risk of ALL in Hispanic Latinos, including IKZF1 and ARID5B and GATA3. Now for something completely different. So I'd mentioned the natural history of childhood leukemia. Twin studies had demonstrated the in utero origins of several subtypes of both ALL and AML. So as I said, this was supported by the same exact gene fusion, which we think supports transfer from one twin to the other through the placenta. And I'd mentioned that several subtypes have been backtracked. And we also know that the ETV6 RUNX1 fusion has actually been found in up to 5% or maybe more of healthy or unselected newborns, which again supports that the pre-leukemia itself is not is necessary but not sufficient to cause leukemia, but also raises the possibility of newborn screening. So to understand which leukemia subtypes first develop prenatally, we think will be essential for any screening and future prevention efforts. For ALL, we see about 75% of the subtypes are driven by gene fusion events, and it's about 63% for AML. Some of these subtypes have been backtracked, and some of them haven't. This is showing, I think, a T-SNE plot of RNA sequencing data from ALL tumor samples, showing the clustering across different subtypes. And I'm just showing you this to highlight the subtypes that have not previously been backtracked to birth. Say, for example, PH-like, PAX5-ALT, and some of these also more recently described fusions, such as in MEFD2 and DUX4. And many of these are actually high-risk subtypes as well. So there's clearly some outstanding questions about the early life origins of childhood leukemia. What subtypes arise prenatally? What is the cell or cells of origin of pre-leukemic clones and across subtypes? So most studies looking into the cell of origin have largely inferred this from the characteristics of the tumor, of the leukemia cells themselves. And also, are some newborns more prone to developing pre-leukemic clones than others? And we think these questions are essential to answer for possible prevention of childhood leukemia in the future. So for those of you who might be involved in the Children's Oncology Group and Project Every Child, this is the registration form. And question 5 here, I believe, was added by Dr. Spector here. Was cord blood banked at birth? And so we were chatting at a COG meeting, I think just before COVID, about how might we use these data on cord blood for potentially investigating the early life origins of leukemia. So that led us to our RECORD study, which I was not involved in designing the logo, but I'm still proud of it because it kind of looks like a record player, although it's like a microscope looking at cord blood. So we developed this to really lay the groundwork for some of these ideas. So aim one was to identify the presence and frequency of prenatal leukemia initiating lesions at birth. Aim two is to try and determine the cell of origin across subtypes and look at the gene expression changes from pre-leukemia to overt leukemia at diagnosis, and whether some of the secondary mutations that we assume occur postnatally might also occur prenatally. And if we can enroll enough patients, can we do some epidemiology, determine whether there might be risk factors associated with the presence and clonal frequency of pre-leukemia. This is what a typical cord blood banked in the US looks like. So we've got this 20 ml portion, this 5 ml portion, and what we call pigtails of about 1 ml each. So keep these cord blood sections in mind. And our original idea was to enroll up to 200 childhood leukemia patients. This has turned out to be, I think, a gross overestimation of the number of patients with cord blood where we could actually get the cord blood, because it turns out that many of the banked cord bloods are in public banks, and they throw away probably over half of the cord bloods that are banked, unfortunately. So even where a parent may have banked the cord blood, unfortunately, we look for it and it's not there. But that was the original idea. And so in my lab at USC, we're collecting the tumor samples, doing whole genome sequencing and RNA sequencing to try and identify the driving lesions. And then we're designing patient-specific assays. These are digital PCR assays where we can, ultimately in the cord blood, we can use these assays to find these lesions at very high resolution. So we kind of call this an artisanal or bespoke study where we're designing these assays on a patient-by-patient basis. So in our lab, we're making sure that these assays work in the tumor, and then we have very good separation to reduce potential contamination, because the cord bloods are being sent to our collaborators in Oxford, and then they are ordering the same digital PCR assays to look for the lesions, the pre-leukemic lesions in the cord blood. And they'll be flow sorting the cells and then looking for pre-leukemia in the different cell compartments. And of the patients that are positive for the digital PCR, they will then be running what's called target-seq, where they can look at simultaneous RNA sequencing and mutation analysis. So they'll be able to look at the gene expression patterns at the single cell level in cells that carry the pre-leukemic fusion. And, some of the kind of important questions that this will answer is which subtypes arise in utero, and hopefully we can look at, as I've said, risk factors that are associated with pre-leukemia. And we see this as a possible pathway to prevention. We like to have lofty goals. I'll come back to that in a moment. So one of the goals is to inform the feasibility of newborn screening. As I've said, we need to know which subtypes arise prenatally. Also, what's the clonal frequency in cord blood? Can we even detect these? And if they can be detected in bulk cord blood or even in newborn dried blood spots. So for some of the patients, we're also collecting dried blood spots, and we will be testing, looking for pre-leukemia, both in their cord blood and in their blood spot. And then we can kind of try and correlate, or at least look at the clonal fraction of pre-leukemia in these two different sample types. And also to shed light on the origins of childhood leukemia, potentially inform novel therapeutics by shedding light on the cell of origin across leukemia subtypes and characterizing the pre-leukemic cells, and also identifying whether there might be key stages for leukemogenesis by comparing the pre-leukemia to the leukemia at diagnosis. And also to inform future large-scale case control epidemiology studies of newborns with circulating pre-leukemic clones. And Doctors Heather Nelson and Erin Marquardt are actually leading such a study, specifically looking at the ETV6 RUNX1 fusion. So our lab is not working on developing a prevention strategy, but others are. So Sir Mel Greaves has proposed some sort of probiotic that you might be able to give to children to help their immune systems develop normally. We think it's probably not ethical, or at least would not be accepted by some parents, right? We saw what happened with COVID vaccines, for example. But maybe by using genetic risk and screening for pre-leukemia, you might be able to identify children at the highest risk of developing leukemia that you could then give some sort of intervention to. This commentary was published a couple of weeks ago in Blood, which I think is a little premature, saying childhood leukemia prevention within reach. This was a study showing they gave mice cytomegalovirus, I think, in the first week or two of life, and it reduced leukemia burden in those mice. So yeah, I think that was a bit of a stretch. But anyway, just to show you, other people are talking about prevention of childhood leukemia. And we wrote a perspective on our record study and kind of the current state of backtracking that was published in Leukemia Journal earlier this year. So where are we at? We've so far consented 54 patients. Of these, they have 23 of these, we've collected the cord blood samples. I say we, these are being collected in Logan's lab, and shout out to Michelle Rosler, if she's here or on Zoom, taking her son to college. Well, she's been instrumental in enrolling patients and identifying the cord blood banks and how to get the cord bloods, and convince parents to, part with. Some of these parents have been paying good money to bank these cord bloods. And we tell them, there might be pre-leukemia in those cord blood samples that you might not want to give back to that child or another child. So anyway, of these 54 patients, we've sequenced about 30 tumors so far. Of those, we've sequenced the cord blood in 20 of those. So we have 20 tumor normal pairs. And we've started testing the first set of digital PCR assays. So I don't have a ton of results to show you today. So this was more kind of presenting our study and telling you the plans for it. But so far, we've successfully tested the digital PCR assay in the tumors for these 4 patients. So we wanted to start with ones that we think we should find the pre-leukemia at birth. So that's ETV6 RUNX1 and MLL rearranged or KMT2A rearranged leukemia. So the cord bloods for these samples, for these patients, have been sent to our collaborators in Oxford. And I think this week they're going to be testing the first ETV6 RUNX1 patient in the cord blood with this assay, which might explain why I was up most of the night last night and couldn't get to sleep. I'm joking. Two additional patients which we're excited about. One has an NPM1 mutation. This is childhood AML. And another has a fusion in the CEBPA gene fused with an immunoglobulin locus. So we're at the stage for that where we've designed PCR primers, and we're going to start testing those soon. And then some of these others, the short read sequencing wasn't kind of definitive. So we're doing long read sequencing for 3 of these. And so this is 14 patients. We have 6 additional patients where we've just received back the sequencing data. So I'm hoping by the time we meet next month, we'll have some more results on what might be the driving lesion. And just to quickly show some of the digital PCR data, this is a serial dilution of tumor DNA and then tumor spiked into control DNA using an ETV6 RUNX1 probe and a control probe down here. So we see very nice separation. And actually, I didn't show this, but when we multiplex the FAM probe, the ETV6 probe with the control probe, it still looks pretty good. Good enough, we think, to move to the cord blood. Okay. Any questions on that before I move to the last part? Okay, something very different again. So I've been interested in a while in how we might be able to use tumor mutations to understand the etiology of childhood leukemia. So this is a story about some of our findings related to tobacco smoke exposure. So I've talked about newborn screening as a possible preventive measure, but if we can identify environmental exposures that increase leukemia risk, then that could be another route to childhood cancer or childhood leukemia prevention. So one such early life exposure is smoking during pregnancy and early life. But case control studies had actually found conflicting evidence, in particular between maternal smoking and childhood ALL risk. And actually, the most recent studies have shown pretty null findings. Now, those studies were largely based on parent interview data, so might be prone to recall bias. But we had previously found an association in a case-only analysis between maternal smoking during pregnancy and the frequency of gene deletions in ALL patients. So initially, this was done back when I was doing lab work using this MLPA, multiplex ligation-dependent probe amplification assay that was targeting 8 known ALL driver genes. And we had interview data in about 560 patients, parent interview data. And then we also had an epigenetic biomarker data in about 200 of these patients. So what we found, both with the interview data and then kind of validated with the epigenetic biomarker, was higher exposure to prenatal or to maternal smoking during pregnancy, associated with a higher number of deletions in the patient tumors. And we then expanded our study using more methylation array data, again looking at this AHRR CPG, also with a polyepigenetic smoking score based on 28 CPGs, and found the same finding. More smoking during pregnancy was associated with gene deletions. Now, I thought this was possibly a chance finding. It still might be a chance finding. But kind of interestingly, about 25 years ago, there was this paper in Nature Medicine from Barry Finette that showed that mothers who were exposed to tobacco smoke during pregnancy, the lymphocytes in their newborns showed a higher proportion of RAG recombination-mediated deletions than mothers with no exposure. You can see in this plot here. So very briefly, what is RAG or VDJ recombination? It's a process in our immune system that helps to generate antibody diversity by inducing DNA double-strand breaks and recombining the variable V diversity and joining gene segments. And the RAG complex recognizes these specific recombination signal sequences, or RSS, at immunoglobulin and T-cell receptor genes. And I'll come back to RSS in a few slides. So importantly, off-target or illegitimate RAG recombination has long been known as a driver of deletions in the progression of childhood ALL, including in both B-cell and T-cell ALL. So given that the prior report in cord blood lymphocytes in mothers exposed to tobacco smoke during pregnancy, and given our finding of this association between increased exposure to tobacco smoke and gene deletion frequency in ALL patients, we were interested in this question: is the early life tobacco smoke exposure associated with specifically RAG recombination-mediated deletions in childhood ALL? So to address this, at least for now, we performed whole genome sequencing in 35 tumor normal pairs from childhood ALL patients with either high or low tobacco exposure. And if we have time at the end, I can show you how we did that, how we separated those, but it was based on the epigenetic biomarkers that I've mentioned. Using the sequencing data, we then assessed the frequency of somatic alterations in the two groups, and then examined evidence of RAG recombination at deletion breakpoints and looked for differences between the tobacco exposure groups. And we also have analyzed mutational signatures in the high versus low exposure patients. So what did we find? First of all, we did indeed find a significantly higher number of genome-wide total structural variants, deletions, duplications, inversions, and translocations, higher in the high tobacco exposure group compared with the low exposure group. So this possibly suggests general genomic instability, but when we looked at ALL driver genes, it seems to be limited to deletions. And this is just an oncoplot to show that the structural variants that we identified overlap the known ALL driver genes such as CDKN2A, IKZF1, etc. We also analyzed presence of these RSS motifs at deletion breakpoints using this FIMO, or Find Individual Motif Occurrences. And so the RAG motif can either be So you have a heptamer, this CACA-GTG, and then this nonamer. They can be separated either by a 12 base pair spacer or a 23 base pair spacer. And this is just data from our deletions, showing the consensus sequence based on the breakpoints in our data. So it's the off-target deletions that we're interested in. We found the full RSS motif in at least one breakpoint sequence of a deletion, and we called that putatively RAG-mediated. So we found this in 93% of deletions at on-target regions, so at the IG-TCR regions, and in about 35% of the off-target regions, or non-IG-TCR. So then we compared both the number and the proportion of deletions with the RAG motif. And just jumping to the proportion, we do see significantly higher proportion of deletions with the full RAG motif at least one breakpoint and at both breakpoints in the high tobacco exposure group versus the low exposure group. And then next, we look for evidence that these are likely true RAG-mediated events. So we saw a decay in the motif signal with distance from the breakpoints, which is supportive. We found that the RSS motif was largely internal to deletion breakpoints. And of 98% of the heptamers were in the correct orientation for typical RAG recombination. Next, we conducted a multi-level model analysis. What this basically showed was that off-target deletions in the high exposure patients had about a 2.4-fold higher odds of being putatively RAG-mediated, that is the RSS motif at one or both breakpoints. And deletions in the high exposure group were almost 5-fold more likely to have evidence of RAG recombination at both breakpoints. And adjusting for age at diagnosis and ethnicity saw some attenuation, but not much. And just finally, we looked at mutational signatures using both SIG profile extractor and looking for de novo signatures, and did not find any differences between the two exposure groups, suggesting that the tobacco exposure is not directly mutagenic, like we see in, for example, lung cancer, but maybe having an effect on the immune system. So we did find 2 patients with the APOBEC signature in the high exposure group, but obviously the numbers are too small to draw any conclusions. So to summarize, smoking during pregnancy may be bad for a child's health. In all seriousness, we've shown that high prenatal tobacco smoke exposure may be associated or is associated with an increased frequency of deletions genome-wide in childhood ALL patients. High tobacco exposure is associated with increased off-target RAG recombination, a known driver of leukemia-forming deletions, suggesting a potential causal role for tobacco exposure in childhood ALL. So some kind of outstanding questions, next steps. Can we confirm these findings in a larger set of patients? Can we determine the critical windows of exposure, prenatal versus postnatal? So I talked before about how, in terms of the natural history of ALL, the second hit mutations and deletions typically occur or are thought to occur postnatally. So how might prenatal tobacco exposure play a role in that? We think possibly if a child is being exposed to tobacco smoke during pregnancy, that may continue or is likely to continue into their childhood. But that's obviously just speculation. Are there other exposures? What we're measuring may perhaps be a proxy for other exposures that we haven't looked at, such as air pollution. And are there other exposures associated with RAG recombination and mutational signatures in childhood ALL? And this is kind of an interest of myself and others in the childhood leukemia space. I think one of the next steps really is to do a comprehensive epidemiology study of mutational signatures, as well as other patterns of mutations in childhood ALL and other childhood cancers. So can we identify risk factors of somatic alterations through association studies? So for example, are there germline variants associated with particular mutational signatures? Are there individual level and area level exposures? And Logan and I have started talking about this and we'll continue to talk about this this week. So just to finish on this point, there have been a couple of studies in adult cancer that have begun to look at the epidemiology of mutational signatures, in particular by IARC. They've looked at mutational signatures in relation to geographical variation in, I think, esophageal cancer and kidney cancer. But in childhood cancer, most epidemiology studies lack tumor samples or sequencing data, and most of the childhood cancer sequencing studies lack epidemiological data. So this may be where, for example, Children's Oncology Group may be helpful. So just to finish up, a kind of summary of my lab's research program and some things I didn't have time to talk about. So all of this, I put together this abstract for a grant a couple of years ago, but I think it kind of neatly shows what I'm interested in, which is, can we understand the genetic risk of ALL? Can we try and figure out the early life origins across leukemia subtypes, and then combine these data to inform the possibility of prenatal screening and identifying children with the highest risk of developing leukemia? Some other things I'm interested in is the genetic epidemiology of Down syndrome, ALL, as well as transient myeloid leukemia and Down syndrome. And with that, I'd like to finish and thank my many mentors and collaborators, and just to shout out that Logan was awarded Mentor of the Year at Children's Oncology Group, and I look forward to celebrating with him on that, and I will happily take any questions. Thank you.
I've got microphones for anyone who has a question. Raise your hand.
So a really interesting talk, Adam. My question is about the first part with the Ikaros variant SNPs that you'd found. And it seems like it's a really high frequency in this indigenous American population. So I'm wondering, has any potential benefit of this variant been found? So in particular, obviously it's detrimental for ALL, but Ikaros also plays an important role in interacting with, for example, the transcription factor FOXP3 to drive appropriate regulatory T cell function, which prevents autoimmune disease. So given the high prevalence, has there been any association of this perhaps with autoimmunity or reduced autoimmunity, which might account for the high frequency?
Really interesting question. So I was halfway through a question, I was going to go off in another direction, which I'll come back to. But the IKZF1 SNP in the promoter region, the risk allele for leukemia is associated with reduced risk of type 1 diabetes and possibly other autoimmune conditions that haven't been looked at.
So potentially, yes.
So potentially, yes. The other kind of hypothesis that we had is that, why was this allele selected in the Americas? We know that, several hundred years ago, the Europeans colonized the Americas and brought over all of these viruses that wiped out, I think, 90% of indigenous populations. So there may have been some selective advantage to immune responses to those infections. We don't have any evidence of that yet, but I think it's kind of a nice hypothesis. And we want to test evidence of selection across other ALL risk alleles.
But you're seeing this even in individuals that were from 12,000 years ago, right? I guess we don't know the prevalence.
The allele is there, but we don't know when the selection started.
Yeah.
Yeah.
Okay, thanks.
That was a great talk, Adam. So many questions, but I'm going to actually just ask one to follow from what Mike was saying. I was at the Aspen Ideas Festival this past summer, and there was a really interesting talk from a woman that's looking at the prevalence of autoimmunity in developed societies versus underdeveloped societies. And she comes from the world of parasitism. And she identified this really interesting primordial hormone that kind of like a language that primordial worms used and has evolved. So now each worm has its own version of this hormonal language. And it is an immune modulator in multiple forms. And so her hypothesis was really interesting, that we co-evolved with parasites over millions of years, and the parasite was an immune regulator. And that when you eliminate the parasite, you essentially set the stage for autoimmune disease, because that immune regulator that's been around for millions of years is suddenly absent, and it doesn't, it can't dampen these abnormal immune responses. So I wonder if you can think about, going back to the idea of the viruses, which is only 500 or, whatever, 600 years ago, that perhaps we really need to think about going back to things that were established long before and think about, do we see the same risk of ALL in the United States versus less developed regions? And your Mexico study might be really interesting because there's very little healthcare in the rural areas. And so you might have a tremendous underrepresentation of cases, but the question is whether they're not happening because there's really still some factor that's present that's protective, or whether that's just because there's no healthcare outside of Mexico City and the bigger cities. But anyway, I think it's this idea of how the SNP would have become established in the context of potential childhood risk, which you should be able, there should be selection for, is really interesting.
Yeah, it's a fascinating point. Kind of a different twist on the hygiene hypothesis.
Yes.
Yeah.
I have a question. Do you know anything about homozygosity for the SNP versus heterozygosity for the SNP within the population? I know that people at LA Kids, for example, have done some mapping of regions of homozygosity, the ROH, by SNP arrays, and have shown a very different distribution of ROH in the Latino populations. And I'm wondering if any of those ROH regions might overlap the region on 7 that contains Ikaros, and whether or not homozygosity might be a higher risk, for example, than heterozygosity for the SNP.
That's a really good point. And yeah, something I wasn't aware of was going on. Did you say it at CHLA? Just down the road from me. I will say though that we know that there's the SNP2 that we called it is on the other haplotype. And so looking at the risk of being homozygote for SNP3 is complicated by the lack of the other risk allele, if that makes sense. Or at least, yeah, I'd have to think about that some more. And how many cases would we actually find that are homozygous at that region? Yeah, a great point.
Are there any other questions? We don't have any coming from online, so let's say thank you again for a great talk.